everyone, in this video we will talk about the recombinant DNA technology. First of all, what is recombinant DNA technology? Recombinant DNA technology is actually the problems which are related to the human genome. Whenever there is a problems in human genome, then the genetic engineer solves these problems through a technology which is known as recombinant DNA technology. Same like whenever there is an issue in our car or in our vehicle, then we go to the mechanical engineers to solve this issue. Same whenever there is a problems in the human genome, we go to the genetic engineers, or the genetic engineers solve these human genome problems through a technology which is known as recombinant DNA technology. So how we will define the recombinant DNA technology? Recombinant DNA technology is defined as that the joining together of a DNA molecules from different organisms and inserting into a host organisms to produce new genetic combinations. Now there is a term which is known as cloning. Cloning is actually the duplications or the multiplication of the human genome. When we have any problems in our human genes like insulin genes, we have a problem, then we clone or we duplicate these genes into a multiple copies. We have a big picture of recombinant DNA technology here, that the first, that the genes to be cloned. We have any genes which are not working properly, then that genes to be cloned. Then we will amplify the gene, means that we will develop the multiple copies of that gene, which is known as amplification of that gene, and at last we have an extract DNA or product which are formed to us through this technology. And actually these three processes are occurs in the recombinant DNA technology. What are the goals of recombinant DNA technology? So the first one is that isolation and characteristics of a genes. Isolation and characteristics of a genes. The second one is that the desired modification in isolated genes. The third one is artificial synthesis of a new genes, modification of organism's genome, interpretations of hereditary diseases and related cures, and at last there is enhancement of human genome occurs through these recombinant DNA technology. What are the tools which are used in the recombinant DNA technology? Or in other words, we can say that tools in recombinant DNA technology. So the first one is that the gene of interest. In gene of interest, we have isolate a genes from a chromosomes, or we can synthesize a gene by a chemicals in a laboratory, or we can synthesize it from a messenger RNA, like messenger RNA, through an enzyme which is known as reverse transcriptase, to form a complement DNA, and from complement DNA we can get a desired gene. So the first step is that the gene of interest, which are tools in the recombinant DNA technology. So the gene of interest in which we can isolate a genes from a chromosome. The second tool in the recombinant DNA technology is the restriction enzyme. There is enzyme which is known as reverse endonuclease enzyme. This is the molecular scissor which can cut the DNA at a specific site, which is known as restriction sites. 6 or 4 nucleotides are arranged in a reverse order in a DNA, and this is known as palindromic sequence. Like we have an example here, that there is a chromosomes, and on a chromosomes we have a nucleotide, like that, like in that order, like GAATTC. And if these are arranged in a reverse orders, like in such a way that below the guanine there is a cytosine, and thymine, adenine, and guanine. So this is known as the palindromic sequence, in which a nucleotide are arranged in a reverse order. So when we cut this palindromic sequence in a that manners like as shown in the pictures. This cut is known as staggered cut. Okay, so like after the staggered cut, the nucleotide are shown as that the guanines are shown on the upper region, and and all the other nucleotide arranged in the below region. So this end is known as the sticky end, in which other nucleotides are attached to the sticky end. So this cut is known as staggered cut, in which after the guanine many other nucleotides are attached to it. So this end is known as sticky end, and this cut is known as the staggered cut. We have another cut which is known as blunt cut, which are shown as that the guanines and cytosines, guanine, cytosine, and below is that in a reverse order. But here the nucleotide are in 4 in numbers, so these nucleotides are arranged in a reverse order. But if we cut this nucleotide as shown in the picture, as shown on the page, so this cut is known as the blunt cut. So we have a two cuts, staggered cut and the blunt cut. But this is actually occurs to an enzyme which is known as reverse endonuclease enzyme, which is a molecular scissor which can cut the chromosome at the specific site, which is known as restriction site. And these nucleotides must have in a reverse order, and this is known as the palindromic sequence. Okay, we have a two cut, that is the one is the staggered cut and the other one is the blunt cut. And the nucleotide which are attached to the to the cut end, this is known as the sticky end. Okay, so we have staggered cut and blunt cut, but actually these all cut are taking place to an enzyme which is known as reverse endonuclease enzyme. The third tool is known as molecular carrier vector. So here molecular carrier vector may be a plasmid, which is the most famous one is the plasmid, yeast and lambda phage. So what is plasmid? How plasmid are used as a vector? So we can explain here that plasmid is an extra chromosomal circular DNA. What is plasmid? Plasmid is an extra chromosomal circular DNA, like as shown in the picture. This is plasmid and this is the extra chromosomal circular DNA. So what why and here is a here question is arise, why plasmid are used as a vector in the recombinant DNA technology? So the answer is that the plasmid have many replication site. The first one is that they have a many replication site. Then they have a resistance gene, and then they have a many cloning sites. So that's why plasmid are used as a vector due to their specific characteristics. Like we have here a plasmid which are shown as in a picture. There is a cloning site, there is a replication site, and these are the resistance gene. There are two types of plasmid. The one is the PSC101, and why they are differentiated into a two types, that is on the basis of the resistance gene. Here the antibody is known as ampicillin in the PSC101. So that is the one type, that is PSC101, and the other one is the PBR322. And in PBR322 there is a resistance gene or antibodies which is known as tetracycle ampicillin. So that's why plasmid are of two types. So the one is the PSC101 and the other one is the PBR322, and they are differentiate. Why they are differentiated into two types? That is based upon the resistance gene. Okay. Now what is now after when we cut the DNA at a specific site, and after that when we cut the DNA and the plasmid, when we insert into a plasmid and recombinant DNA are formed, then after all these we can insert it into the host organisms. So the recombinant DNA which are formed are introduced into the host organisms. So how we can introduce into the host organism? So there are a number of ways that some are the micro, some genetic engineers are inserted it into the host organism through a micro injection or gene gun or alternate cooling and heating use of calcium ion. So there are a number of ways that the recombinant DNA are inserted into the host organisms. Like here, whenever we are inserted plasmid into the host organism or bacteria, so we can treat it into the calcium chloride. Now we diagrammatically explain that actually what the process occur in the recombinant DNA technology. So whenever there is a problems in the human genes, like in insulin gene which are not working properly, and how the genetic engineers manage this problem. So here there is a gene donor, means that the genes are isolated here, and the other in the other side there is a bacteria and plasmid are isolated from the plasmid donor, means that the bacteria plasmid are isolated here. So both are combined through an enzyme which is known as DNA ligase, and then recombinant DNA molecules are formed. So now the recombinant DNA are inserted into the host organism. Like in bacteria, we can insert it the recombinant DNA into the bacteria. So that is that when the recombinant DNA are inserted into the bacteria, this is known as the transformation. And after and after insertion there will be a transcription, translation, and at the end there will be a protein product of human insulin which are not working properly. We can manage this because we can get a protein product through host organisms. Here whenever the host organisms replicate, then the desired genes which are not working properly the genetic engineers can manage in that way. So steps are shown here. The first step is isolation of genetic material by an enzyme which is known as lysozyme. So through this enzyme we can isolate the genetic material. After this we can cut the gene at the recognition site. When the genetic material isolated, so we can cut the gene at the recognition site. Okay, so in the third step we can amplifying the gene copies. When we cut the gene, so we can amplify that gene into a multiple copies. Okay, in the third step, when the recombinant DNA molecule are formed through an enzyme which is known as DNA ligase. So in fourth step, ligation of DNA molecules occur, and at last step, insertion of recombinant DNA into a host, okay, which is known as transformation. When recombinant DNA are formed, so we can insert it into the host organisms like in bacteria, and this is known as transformation.